Yeah, so thanks very much, all of the organizers, three of whom I'm meeting for the first time. And thank you, all of you, for after yesterday's banquet, braving this to lunch. Okay, so um, as Leelati said, I've been talking to her for quite a while, and her fingerprints are all over a lot of what I'm going to talk about. Also, the deep learning part I'll get to in about 15 minutes, and it's something very new to me. So I thought since Gautam's talk in the morning was not, it was a very nice pedagogical talk, but not related really to our talk. So there is some pedagogical component to this talk also. So let's start off with a similar cartoon to what Leopold showed earlier, but I stole mine from Wikipedia. And so this is like the complexity of eukaryotic gene regulation. So here they show a gene here. This is the promoter region that you heard about, and the RNA polymerase typically binds here and transcribes the gene. You have chemical modifications like methylation, which silence parts of DNA. You have a protein called CTCF, which plays a bunch of different roles, but in particular, it acts as an insulator. You have regions quite far away from the promoter called enhancers, which can still turn on the gene because they loop around in 3D space and come close to the promoter. And so all this kind of stuff is there. So the whole game of studying transcriptional regulation has been to try to identify either via prior data, experimental data, and so on, or de novo, possible binding sites for these transcription factors in promoters and enhancers, and various higher order tasks like identifying enhancers and combining other kinds of data sets like ataxia, open chromatin, and so on. But at the basic level, that's what it is. And as you heard, the binding sites for, so these proteins bind to short regions which are not exact strings, but they have a pattern to them, and we call them motifs, and they are modeled by position weight matrices, as we described in the last talk. So it's basically a column-wise categorical distribution of our Goyal matrix. At each position n, you have an independent distribution for each nucleotide alpha. For example, so this would be the algorithm to, this was how you would construct a position weight matrix, assuming that each column is distributed independently via categorical distribution. You collect a bunch of known binding sites, say these are these 6, and you count the number at each position, and then you can show using this Dirichlet prior machinery that the likelihood of seeing any nucleotide at any position is given by this object, where there is a parameter C, which is commonly chosen to be 1. So 1 is called the Laplace add-1 rule, and it is correct if you assume complete ignorance of the prior, and also if all samples are independent and identically distributed. So this is ignoring the M index, which is the position index, is a common formula for distributions of 4 nucleotides. And so you would construct it from these particular sequences by counting the number of nucleotides, adding this number and normalizing it, and that's your final position weight matrix. And as you also heard, you would visualize it using a sequence logo. In this case, you could choose to either normalize the matrix or not, I mean, add a pseudo count or not add a pseudo count. I've not normalized it, but you should normalize it also. And typically, sequence logos are done without a pseudo count just to make them look a bit sharper. Because so the relative heights in each position are the relative frequencies of each nucleotide. And in this case, it is all C. In this case, there is a little bit of T, G, and A. And the total height is the information content, which is given by this. It is basically 2 minus the Shannon entropy. So the Shannon entropy is 2 bits if you have an equal distribution, and it is 0 bits if one nucleotide dominates. And we want kind of the opposite of that. We want to emphasize the ones that dominate, so it's 2 minus that. And so the total height of C here is 2 bits, and here this column is quite a bit less than that. So that's the basic idea. And so there's a one-to-one mapping between sequence logos and position weight matrices. And you can use these position weight matrices to calculate likelihoods of short sequences. So supposing your position weight matrix has a length L, like 7 in the previous example, and you have a string, a sequence also of length 7, then the likelihood that this sequence is explained by this PWM model is just the product of the individual probabilities. So for example, for the sequence CACGATA, which does not appear in the set, we will multiply this number C 0.7 times 0.5 times 0.7 times 0.7 and so on, times 0.1 times 0.6. That would be your answer. And you need to compare that to something. It's a product of small numbers, less than 1. So you compare it with a background probability. This is just the individual nucleotide probabilities. You could have higher order background models, Markov models, and so on, but let's stick with this. And then you could use something like a log likelihood ratio, which is the log of the probability that came from the weight matrix model to the probability that came from the background model, which works out to this fairly simple expression. So position weight matrices are documented in many databases such as Jasper, and sometimes you also find log odds matrices, which are basically these numbers. And the basic idea is that given a longer sequence now, like an enhancer or maybe the whole genome, you would scan this matrix along windows of length L, like 7 in the previous example, along the entire sequence and see where this number peaks. And most of the time, this number will be close to 0 because these two numbers will be roughly equal. So the ratio will be like 1. But now and then you might have a peak, and that's where you predict a site. So that all sounds very good. But as you heard, there are a lot of false positives. So this is a figure from more than 20 years ago from a paper that was, this was a paper that talked about how to predict enhancers, and that's the last figure here. But the first figure is basically scanning the weight matrices of bicoid caudal group, hunchback, standard fruit fly transcription factors which play a role in the early embryo development. And their goal is to predict the enhancers for the Eve gene, even skipped. And if you just look at this, there's nothing you can do. You just see hits all over the place. So they use clustering filters and high stringency matches and so on to kind of narrow it down. So today, but despite these limitations, we still depend on position weight matrices. These days, the only thing you can say is that you have much larger databases thanks to technologies like ChIP-seq, from where you can get these matrices. So with that short recap of PWMs, let's move on. What is the problem with PWMs? One problem is that they assume positional independence. Each column is a separate distribution. And so if you have an example like AA and TT being equally often in the data that you have, then you cannot model that with a single PWM, because such a model will also give equal probabilities for AT and TA because these positions are independent. So one question is, do correlations like this exist? And there have been various literature arguments about whether they exist, or is it good enough to take the PWM approximation. So this, I'm going to talk about initially some work that was published in 2010, and subsequently there have been, so basically this is a paper that talks about extending dinucleotide weight matrices, and I argued that this does better in the data sets of that time. But the reason I'm talking about it is that it also argues that there is signal in your regulatory sequence beyond the core matrix. And that's what we'll be coming to through the rest of the talk. Okay, so a position weight matrix, as we said, is just a multinomial pairwise, position independent count, and it's done by counting the actual number of nucleotides. So that was the position weight matrix with some pseudo count, which we chose to be 1 earlier. And we just generalize it in a very simple-minded way. We take all possible pairs of positions and m within the sequence. So n goes from 1 to 7, m goes from 1 to 7, and you can say that, oh, sorry. So this is M1, M2. So you can assume M2 is greater than M1, and you count all pairs of nucleotides at all pairs of positions. This is a completely legitimate thing to do. You have to choose the C carefully. 1 is probably not important, not appropriate here. But you run into another issue, that how do you calculate a likelihood with this now? So with the PWM, the likelihood is just the product of the individual numbers because they're all independent. And here they are all dependent in some complicated way. So what we did was we defined that DWM and we smoothed it with a pseudo, I think we used a Wittenbell kind of thing drawn from linguistics. And we used a kind of Bayesian heuristic to calculate this, but loosely speaking, we calculated we want to calculate P of S given D. And we did P of D given S, P of S, and then we factorized it in some approximate way and so on. And
Just a question.
yeah.
So does that form correspond to any particular generative model?
Good question. I did not think about the, yeah, I would say no, it does not, and that's a drawback. So it's just a heuristic to, maybe one can think of a way to use it to generate a sequence, but it's not something I thought about. I can't answer offhand. But in terms, this is a data set from yeast chip data, which was fairly standard at that time, about I think they did a few dozen transcription factors and they picked 40 of them that had enough binding sites. And we basically split it into a training and testing set, and we train a weight matrix or a dinucleotide matrix or a dinucleotide matrix adding 10 base pair flank on each side. And we found that adding that flank, even though there's nothing visible in the sequence logo, when you align those sequences, it improved the position, the prediction in some cases by quite a lot. And so that was basically the take home from this work, which I'm just. So now we move on to modern technologies. The previous one was chip, which is hybridizing these proteins on a the transcripts on a microarray and so on. So instead, these days, you just sequence them, and you heard about all that already from the talk earlier. So it's been, I think the ENCODE project started in about 2012. Now there are new variants like ChipExo and so on. So in theory, you get this nice set of peaks in the genome and you look in there and you should see a motif for the protein that you're looking at. And what is the problem here? One is actually, if you don't know the motif, it is not documented. Typically, you would use a de novo motif finder like MEME or the Gibbs sampler, but these data sets are just too large. Those programs don't scale. So MEME, for example, trains it on a subset of like the 500 best peaks and then goes with that. Similarly, you have this problem of indirect binding and mixtures of motifs and so on that you heard about. And at the time, this was like 2016 when I started working on this, there were some ChIP-seq oriented motif finders, but like Chipmunk in particular, which we benchmarked against, but there was a lot of room for improvement. So what I'm talking about next is, okay, so before we get to that, here is a nice paper from 2014, which talks about the phenomenon of seeing motifs for other factors in ChIP-seq data sets that are not for the factor you're looking at. And they particularly identified CTCF, ETS1, and 3 or 4 other factors that have ubiquitous motifs in data sets in the literature at that time. And these are the possible models that they described for seeing why and they call those extraneous motifs zingers because they're quite common. It's not a random motif. There are certain factors that appear a lot. And this can occur for a couple of reasons. Here is an example where you're chipping for blue and your DNA is actually linked here. So when you pull it down, you pull down both this strand and this strand. And therefore, you will get your blue chip, but you'll get this as an extraneous component. Similarly, in this case, they are like interacting in cis, and so you'll pull down this which has nothing, and you'll pull down this which has 2 motifs. You might have indirect interactions where what was called piggyback in the last round. So the blue protein is not actually directly binding the DNA, and in this case, you get a pink motif and a brown motif here, or you get a pink and a brown in the same sequence here. You also have non-specific binding, and that's meaning the blue is not here at all, but you're, for some reason, your antibodies pull this down. And this is, all this is quite well known even at that time. So so what you're dealing with now is a collection of chip-seq peaks, some of which will have the motif that you want. Some of them will have some mixture of other motifs, and some of them might have nothing meaningful. So how do you deal with that? So the starting point was, so this is a paper that came out in 2018, but the starting point for this paper was actually talking to Lilavati about NPLB, which is a program for clustering promoters at that time. And we thought, is there a faster way to do it? And this was a sort of heuristic that I came up with, is a kind of iterated k-means that um, I'll describe in the next slide. And it turned out to be fast enough that Lilavati suggested, why don't you actually forget about pre-aligning the sequence. What we were doing at that time is looking for the motif of the factor we're interested in and aligning a bunch of sequences. And then, so it was not actually on full chip-seq data, but looking for variants of a motif. But instead, you could try combining this with a sort of sampling to look at the position and the orientation. And so it's kind of a Gibbs sampler come clustering program together. So Ankit did some of the coding and the analysis, and Snehal did a lot of the data analysis. He was a PhD student in our lab at that time, and she was a project assistant. And so basically that's the idea. We treated the problem as a problem of clustering by sequence similarity. And instead of having a pre-aligned anchor like the Tata box, we were aligning sliding windows in each sequence. So you have a bunch of chip-seq peaks, which are all of different lengths, but we take a window which is typically bigger than the size of a motif. It's about 50, 60 base pairs long. And we look at similarities over those windows. And essentially, this is the algorithm. And I'll skip this flowchart, I think, but what you do is you start with like 2000 sequences in a cluster, you split it. There is a significance criterion to decide whether the split is significant or not, because you can always split a cluster. The scoring here is the log likelihood ratio of all sequences being sampled from the same model in the same cluster versus these being sampled from one model and these from a different model. So you can calculate that. The thing is, you can always get the split to be positive because you can take a given column, suppose it's all random sequence, you put all A's and T's in one cluster and all C's and G's in another cluster, that will give you a positive LLR. So we have a significance test to control for that. And so we iteratively split each cluster. At some point, we find that certain clusters can't be split anymore. So we keep them, we reject the split, that's the red arrow. Eventually, we can't split anything anymore. And these are like convergence steps too, because you might get suboptimal splits in this hierarchical process. So sometimes you end up with a sequence here that really should be here. So at the end, we get a certain number of clusters. And these clusters are, you can think of them as position weight matrices per sequence of much larger size than the typical motif. And so first of all, we did this, we benchmarked this on various programs, including a previous version of diversity. In fact, in synthetic benchmarks, we actually came second to diversity, but we outdid Chipmunk and a bunch of other programs. In terms of speed, this was actually much faster than anything else. Now, in real data, these are the kind of patterns that we see. This is IRF1, REST, FOXA1, and FIA. And on top of each of these, I've shown the Jasper motif. And at the bottom, I've shown what our program picked up. So for example, IRF1 has a canonical motif, which we pick up clearly. But we pick up a variant which has a sort of secondary motif here. And that's pretty common with many other factors. In REST, it is a heterodimer motif, which is well known. We pick up that motif, we pick up like a 1.5 copy of that motif. We pick up just the first half of the motif. And we also pick up the canonical motif with a much larger spacing. And this is something that most motif finders can't do because they don't look at regions that big. It was actually known by people had looked for these two halves separately and found that they sometimes occur with a wider spacing, but we pick that up. And we pick up CTCF in multiple data sets. We pick up SP1 also in multiple data sets. So the main thing is that we pick up features that are quite outside the core motif. So in this case, NFI, we pick up a secondary motif very clearly that's several base pairs away. We pick up generic GC backgrounds which seem to be important and so on. We pick up a whole bunch of new zingers. These are factors that were not mentioned in their paper. For example, the IRF1 motif occurs in all these data sets, and SP2 occurs in all of these, and so on. And REST, for example, we see that spaced version in a couple of instances in SP2 itself. And we also pick up this strange novel motif all over the place, which I don't think it is related to any protein binding. It's probably some artifact of DNA of the type that will come to the next part. So we've been, this was published in 2018. We've been working on it a bit more recently. We use the same general ideas, but using more of an EM algorithm to cluster tabular data with the goal of analyzing things like medical records where you might have a patient data set from a very heterogeneous population. And the original idea, which again came from a project with Lilavati, and she was a sounding board for this paper. And then right now we are looking at rewriting, I mean, this is in progress, and there are some efficiency issues, but hopefully it will come out soon. And Ishitwa, who's a project assistant in my group, has been working on that. Right, so that's about, I think, 1/3 of the talk, and the remaining part, let's talk about neural networks. So I myself, I'm very new to neural networks, and you heard a lot about it from the previous talks. Many speakers have been using them. For those who are not that familiar with it, this is a typical multi-layer perceptron. You have a bunch of inputs in this, the green layer. In this case, you have a single output, which would be the case when you want to do a classification, which is what we're doing here, like you want a yes or no answer. You can also have networks with multiple outputs, and those are when you have a multi-valued classification, or more sophisticatedly, if you want to generate sequences or images and so on, you'll need a more bigger output. But in this case, you have inputs that take real numbers as input values, and then each layer has links to all neurons in the next layer. It's all feed forward. There is no feedback link in this network, and these links all are basically computations of this sort. So they take the inputs, W1, W2, W3 are the weights, X1, X2, X3 are the inputs, and you have a linear function with a bias, sum of Xi, Wi plus B, and it computes that. But in the output, it's also sent through an activation function, which is nonlinear. If you don't have this activation function, then despite all this, what you get at the output is a linear combination of the inputs, and you don't really gain anything. So the nonlinearity is essential, and it turns out that with this setup, you can do very sophisticated calculations. So in the choice of the activation function is important. It has to be nonlinear. In the early days, they chose functions like the sigmoid, which, so here are some activation functions. The sigmoid and tanh have this nice smooth behavior, and intuitively it made sense. It turns out that, first of all, it's slow to calculate. Secondly, the flattening it out at high values is a problem, because the way the neural network is trained is via backpropagation. That means you have when you're training it, you have a known output and you got the measured output, and you're using the difference and propagating it back via basically a gradient descent kind of approach. And so you have to take a bunch of derivatives which don't work when it is flat, so it becomes difficult to train. But also, it turns out that this ReLU, which is the most widely used activation function now, works it's very fast to calculate and it works as well as better. So ReLU basically means that if x is positive, then it is x. If x is negative, then it is 0. And that simple nonlinearity is enough to model quite sophisticated functions. There are many other examples of activation functions. So I think other very common one is leaky ReLU, where you have two different slopes on the positive side and the negative side. So that's the basics of a neural network. And now you want to, so it is basically something that calculates a complicated function. And now in the case of analyzing DNA, your input is a bunch of nucleotides and the output is, it could be a number, it could be something else. Yes, I forgot to say that in the output layer, this output is typically passed through a sigmoid function like this, meaning the output calculates a linear function, which could be anything between minus to plus infinity, but you pass it through a sigmoid to give it a value between 0 and 1 and interpret that as a probability. So that's the common case. Most of these prior networks use convolutional neural networks, which I'll describe in the next slide. And they started off mostly as simple classification problems. So the problem that we will be talking about is, given a sequence of DNA, is it likely to be a binding sequence for your protein of interest, or is it not? And we will use that using, we will address that using ChIP-seq data sets, and for a negative set, we'll use things that is open chromatin but is not bound to ChIP-seq. So here is a cartoon of how a convolutional neural net works. The main difference between this and the multi-layer perceptron I showed earlier is that the filters in these are not, so they are basically computation, which maybe I should use the board for this, or maybe I'll do it here. So essentially what you have is, each of these is a matrix of numbers. Sorry. Let's switch to this. So each of these is a matrix of numbers. Let's say it's like 3 by 3 or 5 by 5 or something. Let's say 3 by 3 for simplicity. And the input is an image which is rasterized. So let's say it's a monochrome image and not a tiger image. But what you get is, let's say it's 16 by 16. So you get 16 vectors of length 16 each, which are all piled together into one vector of length 256. But the filters the network knows how it is structured. And each filter is a square filter in this case with a bunch of numbers. And if it is overlaid over a piece of the image like this, supposing the numbers are like 1, 0, 0, 0, 1, 0, 0, 0, 1, and supposing you overlay it over a piece of the image that has a diagonal line like this, what will happen is that it will pick up that feature. Whereas if you have a piece of the image that has a horizontal line, it will not strongly pick up that feature, only this one will light up. So each filter picks up shapes from the input layer, and that's in the first layer. So in this case, you could say that the last, and remember there's a cartoon, this identifies an ear, this identifies a part of an eye, and so on. And then the next layer of filters combines these in some ways. So this assembles the eye from these three pieces, this assembles the ear from this piece, and so on. And finally, these are combined into an output filter, which tells you, is it a tiger or is it not a tiger? So that's the idea. And in a real CNN for image classification, you would have typically a few dozen filters here, like typically powers of 232 or 64 or something. And there are many layers. You'll also have a kind of layer called a max pool, which I'll come to. And so you might have a layer of filters, another layer of filters, a max pool layer, and it might go on like that. But finally, it turned out to work very well in classifying images. So this is some neural net that we've been working on since about a year now, a bit more than a year. Mohan was a master's student who wanted to do a project with me, and I suggested he work on Dragon, which he pick up Anshul's neural net program, which was for basic classification of sequences. They have a nice tutorial and so on. In fact, Anshul gave a talk, a Zoom talk in this very room 2 years ago, and that's where I got interested in it. So he finished that project and is working somewhere. Divya did some more work on it, and recently Shitwa has also been picking up on it, and in particular, I know various data sets. And Lila is, as always, the sounding board. And a couple of the slides in this talk actually came from her suggestions in this meeting. So this is the structure of this network. So basically, Dragon is it has, I think, 2 layers, but the training version has only 1 layer. And subsequently, they published BPNet, which is a lot more complicated. But Mohan came up with this structure, which was inspired by image processing. And it turned out to be pretty optimal. He has one convolution layer of 32 filters. A second one, he had a doubling sequence, 32, 64, 128, 256. And after every 2 convolution layers, you have a max pool. A max pool, I will describe it next, but it basically crunches up the inputs into a smaller dimension. So where you had 1,000 inputs here, you now have only 125. And the second max pool, after the next 2 layers, crunches it even more. And then you have a flatten and dropout and so on. So you take, in this case, so the input is of shape 1,000 times 4. There are 1,000 nucleotides in our inputs. We take quite long input sequences. And 4 is because there are 4 nucleotides. So we do what's called one-hot encoding. So the input looks like this, where A is encoded as 1000, C as 0100, and so on. The reason for this is that you don't want to encode them as numbers 1234 or 0123 because that implies a value. And this is sort of value neutral way of encoding.
Sorry,
And,
so these are like the color channels, right?
The color channels, I'll come to what they are. And you're saying they're like color channels. They are channels, but they're not really color channels. You could think of them as color channels. A is a color, C is a color, and so on. Yes.
Okay, so they're going into the color, like the 4.
You could say there are 4 colors, A, C, G, and T.
And it's a 11,000 sequence. How is the 1,000 found? Is it the same way, like in a peak and you can?
1,000 was arbitrary choice, but we've been playing with lengths and so on. But what we find, as I'll show, is that there is a lot of signal going up to 1,000, and maybe we should go even further than that in terms of what does this use to classify a sequence. So this is a kind of cartoon of how I think the setup works after quite a lot of playing around with networks and trying to understand how these image networks work and so on. So based on that, each of these, it's called a 1D convolution filter, but it's really 2D. The second dimension is the number of channels. And in this cartoon, we have 3 of them. We will really have 32. And so each of these is a square filter, which works just the way that it does in a convolutional network. And the effect of these filters is if you overlay a filter on any part of the sequence, what we will have is just a product of the individual numbers. Like if you overlay this red filter here, you're basically multiplying this number or adding this number to this number to this number to this number and so on. And that's basically what you do with a position weight matrix. So you could think of these as analogs to a position weight matrix, except that these are not they are not probabilities. They can be negative, they can be more than 1, and so on. So in fact, in the Dragon tutorial, this analogy is made explicit, but they work only with synthetic data where they have planted the motif and they show that they recover the motif. So that's the first filter, the first layer of filters. And the cartoon here is like maybe this first layer identifies patterns in the DNA at certain positions. So the red filter peaks here, here, and here. The green filter peaks here and here. And then the next layer of filters serves to combine these patterns. These filters are 3 times L because there were 3 filters in this layer, therefore 3 channels in this output. In our system, there'll be 32 times L. And so these combine these signals in various ways. So for example, maybe the green filter peaks where it sees red and green here, and so it gives you a peak here and here. And the blue filter peaks where it sees all three together, and that peaks here. And then there's a max pool layer. So what max pool does is it's of length 8 in this example. You slide it along without overlapping, and you pick the maximum values you see in that region. So in this first 8 base pair window, you get a peak, and that's your peak for this max pool. So, so if your max pool filter is of length 8, it's the output is reduces this length by a factor of 8. So it's basically kind of crunching up of the dimensions of the space. And then we have 2 more convolutional filters, 2 more output filters, and that's the basic network structure. So I did a bunch of benchmarks playing with the number of filters in each layer, playing with the number of layers, playing with the dropout rate. There is a dropout layer here, for example, we use 0.2. So the final optimal network is basically this, but it turns out to be very, very close to what Mohan originally came up with. So it's kind of remarkable. And so that's what I'm using for the next slides. So this is, we're going to talk about it as a classification problem. And we're classifying, okay, so here's a comparison with BPNet. So BPNet, we could not get it to run. And there is now a new version called Chrome BPNet, which I haven't tried yet. But this is the architecture as given in the paper. So it's quite a complicated thing, and it has many layers, and it has like connections between multiple layers. But mainly it has this stride thing. Each layer has a stride that doubles from the previous layer. And that's something that again comes from the image literature. So we basically tried re-implementing this in Keras just as a classifier, this architecture. So when I say BPNet in future, I'm talking about our implementation. Most of the older DeepNet programs you are not able to get to run, basically because they depend on earlier versions of TensorFlow and the associated ecosystem, which are no longer even available to download, even if you use a virtual machine or a virtual environment. So we were able to run DeepRAM, and FastSK and LSGKM are not neural nets, but they are K-mer-based programs. So basically what they do is they take K-mers and they train a support vector machine based on them, and they use that as a classifier, loosely speaking. And they are actually pretty good. This is the ClipSeq dataset from the DeepRAM distribution, and we actually outperform DeepRAM on that dataset. So this is like RNA binding. And we also outperform these 2 programs. This is the ChIP-seq dataset from the FastSK distribution. Here we actually do a bit worse than the K-mer-based methods, but the caveat here, which I'll come back to, is that in the classification program, you need a negative set, and their negative set is a scrambled version of the positive set, and this is pretty widespread in the literature. People either use random DNA or they scramble the positive set, and that's the negative set. That's not a biologically interesting problem, in my opinion. So we made a data set of 117 factors. This was done by Ishitwa from the ENCODE database. Here, the positive set and the negative set are both attack-seq open chromatin. The positive set has ChIP-seq peaks, and basically it's each peak shows up in at least 2 replicates from the ENCODE data. And the negative set is open chromatin, where none of the ENCODE ChIP-seq datasets show a peak. So this is for 117 factors, and we outperform in most cases the K-mer-based methods. These 2 programs are both descendants of a program called GKMSVM, and they are, I think, basically the same algorithm, and the performance is essentially identical. We also outperform the 2 neural nets we tested, DeepRAM and our version of BPNet, in some cases quite substantially. So we also looked at, for example, how do you, so this is where you're classifying bound and unbound. But you could also ask, if you have ChIP-seq data for two different factors, can you tell, given a new sequence, which factor is binding to it? So for that, Ishitva created a data set for okay, this is pairwise comparisons of LSGChem and BPNet, showing that they're fairly similar, and LSGChem and FastSK are essentially identical. This is a data set where we have and each of these has 3,000 sequences in the positive and the negative set each. So these are balanced data sets. These are data sets where you have ChIP-seq peaks for each of these 13 factors, which are not shared with any other factors. We just wanted a kind of clean data set. So factor A versus factor B, and can you tell which factor binds? And here, again, we are outperforming both the KMO-based methods and the two deep nets that we've tested so far. In fact, we are essentially nailing most of the classifications in this task. So at this point, we have a very good sequence classifier. And the next question is, what is it seeing in the sequence? Is it just a motif? I haven't shown this here, but if you just look for a motif or no motif and use that as a classifier, you actually do pretty badly. For the reasons that we discussed earlier, that many ChIP-seq peaks don't have a motif in them. Right, so next up, we thought, okay, so what about if you have, so all these guys have a motif in some fraction of the sequences at least. What if you take out the motif? So what we did was, this is data for CTCF and NRF1, we've done it for a few other factors. What we did was we took the Jasper weight matrix and we chose log likelihood ratio threshold such that in the positive set, the bound set, you find a motif instance in 95% of those sequences. That's our threshold. We applied that threshold to mutate all instances of that motif in all the data sets, positive, negative, training, testing, all of them. And so in theory, fairly low stringency match of this motif is gone. And we are still getting an EURC of close to 80% well, 75% in both these cases. So then the question is there something else in that sequence that, so these are 1,000 base pair sequences and our classifier is looking at something where all the motifs are gone and it's still seeing something there. So what is there? So basically we are saying that you remove the face of the tiger, but you're still classifying it as a tiger because of something else. There were also some data sets where we tried to do the same motif removal thing, but there were very few significant motif matches. In fact, if we tried to get the same 95% threshold, the log likelihood came out negative, which basically means you're trying to match things that are worse than random. So these are two examples of that. And again, we are getting, so nothing was mutated here, but these are the scores that we are getting. So this is a sort of block mutation analysis we did. So essentially at every, we mutated 5 base pair windows and slid that window along the sequence. And how much is the score hurt by every 5 base pair window that you mutate? And the heights of these are the change in the score. And the what's plotted underneath that is the actual nucleotides that occur in that sequence. And this is for CTCF. This is the sequence logo for CTCF. And it's, there is no reasonable stringency match to either of these in here. And what we are seeing is that there is stuff quite far away from the center of the sequence that contributes, but also in the center, even though the motif is gone. So what is there? We zoom in on this piece, for example, and we see that it's not a CTCF motif, but you could kind of argue that if you look at this version of the motif, you have a double G and 3 nucleotides, and then you have a G here, which is a T here, and then you have 5 Cs with some mismatches. It's kind of like a CTCF, which probably won't work because of this T not being there. And that's something we see in other factors. This CPB, which is again a case where we did not see significant motif matches. So here you see again contributions from all over the 1,000 base pair region, but of course particularly in the center. And if you zoom in on the center, you don't see a match to this motif, but you see things that kind of sort of look maybe or maybe not. But you see patterns in there. So we could ask generically, so this is like one random sequence out of hundreds that we've analyzed in this way. If you look at all the sequences that show as positive, that are classified as positive and try to see what is there, most of them show this kind of local peak. So what if we take these local peaks and then analyze those for motifs? In fact, the BPNet ecosystem has a program to do exactly that called Modisco. And again, I was not able to run it. It came with some error, so hopefully we'll fix it with Anshu's help at some point. But instead, what we did was ran thickweed on these local motifs, or what they call seclets in the Modisco language. So each of these is a small sequence, expand it a little bit on both sides and run thickweed on it. Okay, so there's a third example of IRF4 where this is the canonical motif according to Jasper, and we just don't see it, but we see all this other stuff. So in IRF4, we took all those little peaks across all the hundreds of positive sequences, the things that it classifies as positive, and clustered them with thickweed. And we find some, I won't call most of these motifs, they are some sort of patterns, but if you look at this one, it is sort of similar to this, but not really. It is the T stretch is a bit longer, and instead of a double G, you have a GTA. And so it's really hard to say what's going on here. Similarly, for CTCF, in the IRF4 case, there was no canonical motif. So this is on the unmutated data set. In CTCF, we have mutated out all the motifs, and now we are seeing what else is there because it's still 75% accurate. And we don't see anything like the canonical motif, of course, but we see these interesting poly G stretches, which, so this piece, for example, occurs in many sequences, and it is quite similar to this part of the CTCF motif. And this also could arguably be kind of similar to, so it's like we find little bits and pieces of. So as I said, this is very much work in progress, but this is kind of where we are at. So the message of this is basically we find signatures of functional sequence, TF binding sequence over a large region, much beyond the core motif. And those signals are informing the classifier something. Whether those signatures are important in vivo, we don't know. It could be evolutionary artifacts because this has evolved to bind this protein, but it's also evolved to be robust to mutations and so on. So often you see other signatures nearby. So for example, some of these could be remnants of old CTCF sites or whatever. And the classifier does well even when the core TF motif is removed. Like we could recognize the tiger when its face is removed, but probably it's not going to function when the core motif is removed. That's something you have to do an experiment to decide. And a possible conclusion from this, if you look at a lot of the other deep learning literature, including AlphaGenome, which came out a couple of weeks ago, their main focus is single nucleotide variants and their functional impact. Like they say we can predict these things at single nucleotide level. And personally, I think it is not at the single nucleotide level, and it is a complex interaction of many things. So it can be a little bit misleading to focus on that. That's my feeling. Going forward.
Can I just ask one question? Yeah. In all these scenarios, like how much data would you get to train this network, for instance?
There is a huge amount of data out there. All the things that I showed, we had 3,000 positive and 3,000 negative, but that's partly because I'm running all this on a display class GPU, which has a limited amount of RAM. So right now we're looking at, I mean, we have other GPUs which I have not set up to run with this yet. And I've also been using Colab a little.
But 3,000.
Yes, but in principle, I think you could scale it a lot. So the goal, and also these are balanced data sets. So I would like to convert this into a predictor across the genome. So you can scan it across the genome and predict possible binding, but that's a highly imbalanced data set. And right now it's not working very well on imbalanced data, but I think it can be done. But the scale, I mean, I think the size of the data set is not, it can scale to thousands. We haven't done that yet. Thousands of sequences of length, 1,000 base pair each.
When you say it cannot scale to larger data sets, you mean?
No, we can.
You can, yeah.
But right now, we can't deal with severely imbalanced data sets. Right. Also, I showed AU ROC curves mostly, which are good for balanced data. For imbalanced data, AU PRC is better. We have those. I didn't have them in the slide. So one of the goals going forward is also to generate data that has, generate sequences that have a desired in vivo function, for example, for binding a TF. Right now, people do that by just implanting the motif and it works. Can the neural net learn to do that? So this is just work from 2 summer students over the last couple of months. They made a GAN, a generative adversarial network, which learns from the same data sets that I showed you to generate similar sequence. And basically how a GAN works is you have a generator and a discriminator, and the generator generates stuff and the discriminator tries to tell whether it is different from the real stuff. And when it is not able to discriminate, then you stop. So in some cases, like for fossil 2, they were actually, so what they did was they ran meme on both the real data and on the synthetic data generated. In both cases, it gave this motif. But in other cases, CTCF, they got like half the motif generated, but not the full motif. And an interesting observation here again was that if you gave it synthetic data where there is a single motif with random background, it didn't really work very well, but it did well with real data. So that again ties in with all bioinformatic, regulatory genomics algorithms tend to assume a random background, but it's not random and it could be important. So the analogy here would be like a motif in a random background is like a tiger in a white noise background, but I don't know whether anyone has done this experiment, but real data sets have actual photographs and maybe that's also important. So that's one future prospect. Another is to apply to other kinds of sequences. So we just started some work with Tom Sexton at Strasbourg on analyzing and designing TAD boundaries. We don't know the likely outcome of that yet. So with that, I'd like to end. I think it's like 10 minutes early, so yeah. I'd like to thank everyone on the slide. I'd like to thank Birak particularly for that GPU computer. It's all running on that, though it was bought for a different purpose.